The overall goal of this procedure is to produce a large area of nanopatterned substrate from small nanopatterned molds by using a simple and affordable yet versatile stitch technique.
This method can help study substrate nanotopographic modulation of cell phenotype and function at cellular and molecular levels, such as expression of focal adhesion proteins.
The main advantage of this technique is that it is simple and cost-effective.
To begin, prepare the siliconized silicon mold in the PDMS pre-polymer as described in the text protocol. Put the siliconized silicon mold in a 60 mm Petri dish. Pour the PDMS pre-polymer on the silicon mold in the Petri dish. Place the Petri dish in a plastic desiccator and degas for about 10 minutes until all bubbles disappear. Transfer the Petri dish to a hot plate and cure the PDMS pre-polymer at 70 degrees Celsius for 4 hours. Carefully peel off the PDMS mold from the silicon mold by using tweezers. Determine the orientation of anisotropic PDMS nanopatterns, such as nanogratings, under an optical microscope and mark it on the back side of the PDMS molds with a marker pen. Clean a silicon substrate with ethanol in a fume hood, and then dry it with compressed air. Trim off the unpatterned areas of each PDMS mold with a blade.
The PDMS molds should be aligned as close as possible, but not touching the surrounding mold to minimize the